Hello, everybody. I think we are live. I'm here with Dr. Eran Stark, who is joining us on this first session of today's program. And he's part of the Emerging Techniques schedule of our conference. And I'm really happy to welcome you here. Sorry for being a bit late. We're experiencing some technical issues, but I hope you understand that. And we'll try to work them out during the session. So I hope this works well. Dr. Stark will talk about spike timing dependent reliability of information transmission and effective connectivity. I think you will appreciate during his talk that he uses really top of the notch, really innovative techniques for doing that. And I will not eat up more of your time, and I will let him to speak about his research himself. I want to just remind you that you can use the chat to write comments. And you can also ask questions using the Ask a Question button. And in the end of the session, we will take these questions and answer the most popular ones. Without further ado, I'm handing over to Dr. Stark.
Okay, so good morning, everybody. I hope that you can hear me properly. We've been having some sound issues in the morning, and I thank everybody for attending. So my name is Eran. I'm from Tel Aviv University in Israel, and I will tell you about spike timing dependent reliability of information transmission and effective connectivity between neurons in freely moving animals, that is mice. This work is work that has been done by 4 talented people in my lab. So I will note their specific contribution during the talk, but the basic point is to say that these are data that we have been recording in the lab for the past 2.5 years. I will discuss information in the brain. Two very elementary questions that come up when you think about the brain and information are the following. One is, how is information transmitted within the brain? How is information from one part transmitted to another part? How is one neuron communicating with another neuron? How is one group of neurons communicating with another group of neurons? So this is one very basic question. And the other, perhaps basic question is, how is information stored in the brain? So we are exposed to various stimuli, we learn, we experience things, and these items remain with us for seconds, minutes, hours, years. How does this happen? So the working hypothesis, that is to say the assumption that I will use throughout this talk is this schematic circuit that I drew here below. Formally, the working assumption or hypothesis is that information is transmitted and stored by networks of spiking neurons connected by synapses. So we have at least 2 critical elements here. One is a neuron and the other is a synapse. Now, this is not a unique hypothesis. This is perhaps a strong working hypothesis that underlies the work of or the thought processes of many scientists. I just want to emphasize that it is not a fact. Information could be transmitted by or stored by other means, which it probably is, at least to some extent, by subcellular compartments, neuroglia, etc. But I will focus on this specific working hypothesis. So the 2 overall broad questions that I described in the beginning can be restated using this working hypothesis. So the first question of how is information transmitted in the brain, we can rephrase it as, how is information transmitted from one neuron or one group of neurons to another set of neurons or another neuron. And the corollary to this question is what makes an individual neuron spike? Now, this is not a new question. As I mentioned, it's a long-standing open question, I think, in systems neuroscience. And I will try to give you our insights about this during the first part of the talk. The second question that I mentioned is how information is stored in the brain, and under the assumption that some types of information, for example, reference or long-term memory, is expressed by the strength of the synaptic connections between neurons, then we can rephrase the question as how does a synaptic connection between two neurons or two sets of neurons change its strength following some experience or learning, etc. So with these more specific questions that again have been addressed a lot of times in the past, I will now focus on the first. I will discuss this and then go to the second. So our first topic will be what makes a neuron spike. And again, it can be rephrased by asking what properties of the input to a cell does the neuron have the largest or highest sensitivity to. And it can be extremely or simplified or rephrased as two options. Yes, of course, they are not black and white. There's many intermediate possibilities, and I will focus on this actually. But at two extremes, one can think about two modes of operation of a neuron. One would be the temporal integration mode, and the other one would be the coincidence detection mode. And this can be illustrated by this review from a couple of decades actually ago, in which the temporal integration mode is essentially an integration or summation over multiple EPSPs or EPSCs that arrive at to the cell. They are integrated at some point in space where the neuron makes the decision whether to spike or not. And if the time constants of the EPSPs are large enough, then they can add up one EPSP, another EPSP, and then another one, and then generate a spike. If, on the other hand, the forgetting factor or the time constant of the EPSP is shorter, then if two EPSPs arrive not at the same time, by the time the second EPSP arrives, the effect of the first EPSP will already be gone. So in this type of a mode of operation, the cell would favor simultaneous arrival of inputs. In other words, if two or more inputs occur at the same time, it will spike. So this would be the coincidence detection mode. And if we try to rephrase these two modes as a set of individual trials, so we take a thought, this is a thought experiment. We take an idealized or a conceptual cell, and we present it with inputs. If it operates in the integration, or we will also call it the rate coding mode, then such inputs which have similar number of spikes at some time constant will at some time window will yield a similar output of the neuron. On the other hand, if the cell is operating in a temporal coding or a coincidence detection mode, then to observe a reliable, consistent output, then the same input would yield a reliable, consistent output, which would look more or less like this. Okay, so each time here would be the time of an output spike. So basically, instead of asking whether the EPSPs have a time constant which is long or short, because that would be only part of the answer, right? The other part is what is the integration time in the cell body. So there are multiple factors which all interact to give the overall behavior. So without knowing every single thing about the neuron, what we could do again in this thought experiment is to apply the same input to a neuron, exactly the same input that would be rich in time. So it would include multiple frequencies and multiple patterns of input. But it would also be corrupted by the specific type of noise that is in the brain. And then we could ask what would be the reliability of the spike train at the output. And we could measure this reliability. We don't know how to measure it perhaps, but we would like to measure the reliability at different time scales. So if the neuron is a temporal integrator, then the time scale at which the reliability would be highest would be relatively long. Whereas if it is a coincidence detector, the time scale at which the reliability of the spike trains would be highest would be shorter. Now, by reliability, what I actually mean is the similarity between spike trains to different inputs, again, in the presence of ongoing noise. And this is a computational problem that has been addressed many times in the past. Here are just 3 or 4 solutions. The approach that we took follows the methodology of Schreiber from 2003, in which what we do, we present each spike train as a point process. So that is a series of delta functions. We convolve each delta function with a Gaussian window, which has some time constant sigma. So the width of the standard deviation of the window dictates the time constant of integration or detection. We obtain a firing rate vector out of the sequence of impulses. And then we we apply this process to each trial I, and then we compute the Pearson correlation, the correlation coefficient between every pair of trials. And our measure of reliability is the average over all of these correlation coefficients. So stated more simply perhaps, our measure of reliability is the correlation coefficient between the smooth spike trains averaged over all pairs of trains. Now, this is just a simulation, a simple demonstration of the idea. So here we have 25 spike trains that we generated by a computer. They're not recorded from the brain. The events themselves were generated at the vertical dashed red lines and jittered a little bit. And then we applied the methodology of computing the reliability. And the standard deviation of the Gaussian is changing from 1 millisecond up to 128 milliseconds. And you can see that the peak of the correlation coefficient or the reliability is around 8 milliseconds, which is similar to the jitter that we induced. So this would be a cell that has high consistency or high reliability of its spike trains at small windows. So we would term it a temporal coder. And this is distinct from this cell, which has highest consistency at longer timescales. So this would be a rate coder. Now, it is trivial to say that if we would increase the standard deviation, this curve would not increase indefinitely. So this is just to say that we have to take all of these types of analysis in perspective of relativity. But within the range of up to 100 milliseconds or so, again, this type of a pattern we would characterize as a temporal coder, and this would be characterized as a rate coder. Okay, so now we are ready to actually go to the brain and do this and apply these ideas. This is just to say that if you jitter at the spike trains at multiple time windows, the reliability profile recovers these time constants rather well. Okay, so what do we need in order to actually do these experiments? First, we need, if we want to apply it to the intact system, we need an intact animal. So we are using mice for our experiments, freely moving mice, because there are sufficiently, they have a sufficiently small generation time for us to be able to use transgenics and transgenic modulations. And on the other hand, they're large enough to carry these large Faraday cages in which we have multiple electrodes and optical elements, and still run around and groom and be sufficiently happy to carry out complex behavioral experiments. So we work with freely moving mice. The other thing we need is multi-site optical control. So why optical and not electrical? Electrical can give us very precise temporal control, but the spatial control and the cell type resolution is better with optogenetic techniques. And we developed hardware to guide light into individual parts of the brain while we record the neuronal activity from these parts. So here is a quick demonstration of the technique. We take a small diode, we couple it with a fiber. We generate a fiber a diode assembly, which looks sort of like this. We take a multi-site silicon probe or a tetrode array, and we couple a single fiber to each shank of the silicon probe. This is all mounted on a movable microdrive, which is cemented to the skull at this point. And then we can apply various wavelengths independently to various locations in the brain. And by making the neurons sensitive to these light waveforms, we can induce activity in different types of cells at different locations. The next thing we need is a pattern of stimulation, which would be identical from trial to trial, and it would also be rich in the frequency content. So we use a filtered white noise pattern. And the last thing is to actually combine everything and record from multiple cells during the application of these white noise patterns. The person that spearheads this project is Amir Levy. So the data that I will show you are mostly recorded by him and some by other people in the lab. So here, what I show you is an example of direct application of white noise to an inhibitory neuron. This is a PV channel rhodopsin animal. So this neuron, which has this type of waveform and this type of autocorrelation, and is actually activated or has a monosynaptic cross-correlation with another cell, which seems like a pyramidal cell. So after these parentheses, I can say that when we apply white noise to this cell, the same white noise, multiple times, we get these types of spike trains. So they're not identical. There are some things that seem similar, perhaps. If we apply the reliability measure, we can see that the thing that is constant or the most reliable aspect of these spike trains is the spike count in windows of approximately 100 milliseconds. So this is a monotonically increasing curve of the reliability as a function of the standard deviation, so we would call this a rate coder. And what we find is that when we apply this type of stimuli to interneurons, that is, we directly activate cells, similarly to what we do or what people do when they inject intracellularly, inject current intracellularly, we receive this type of behavior in most of the cells. So again, this curve can be looked at from above, and we can stack multiple curves of multiple cells that were recorded or manipulated either simultaneously or at different sessions or from different animals together. And they look like this. So most of the curves have a peak at the longest time window or standard deviation of the Gaussian that we tested. A minority behave as the temporal coder or coincidence detector. Most of them behave as rate coders. So this is what happens when we directly activate neurons with a white noise pattern. If we activate the presynaptic cells, so here we activate pyramidal cells. These are CAM-kinase 2 channel rhodopsin animals, in which we apply light to one spot in the brain, and we record the activated cells, but we compute the reliability for a postsynaptic cell that is not directly activated. Here we have multiple repetitions of the same white noise pattern that we apply also to the directly activated cells. And here we observe, when we compute the reliability curve, we observe a peak at short time windows. Now, what is short, it depends whether we do these experiments in the neocortex or the hippocampus. If it is in the hippocampus, the peak is around 4 to 8 milliseconds. If it is in the neocortex, it's actually shorter, it's between 1 or 2 milliseconds, but the vast majority of the cells, it's the same type of cells. It's interneurons that have the waveform of a PV cell, but we don't activate them directly. When we activate them indirectly, the same cells behave as temporal coders. So to go back to the question that I presented to you 20 minutes ago, we can now say that the property that makes a neuron spike differs whether we activate the cell directly or indirectly. If we activate the cell directly, that is, we inject a current, in our case it's a photocurrent, directly to the cell, it acts as a rate coder. So the integration window is large, relatively large. On the other hand, if we activate the cell via its presynaptic peers or its presynaptic counterparts, then it actually behaves as a temporal coder. So the integration window or the standard deviation window for which the reliability is highest is shorter. It's around 1 to 6 or 8 milliseconds. Now I want to emphasize that in the live brain, no cell receives a direct photocurrent. So there are no pipettes or diodes in our brain when we actually carry out our natural life. So the situation that applies to the actual brain is the second situation. Most of the cells, or all of the cells probably, are activated by synapses, except for the very first sensory neurons. So that would be our answer to this type of a question, again, in this type of preparation. The second question that I posed is what changes the synaptic strength. And here again, I can more from a didactic point of view or from a logical, we can phrase or form two possibilities. Again, they're very simplified and they're not mutually exclusive. But one possibility is that a synapse or a synaptic connection between cells would change based on the use of them. So this would be called use-dependent plasticity, and it is often modeled in experiments using high-frequency stimulation, which often induces long-term potentiation, or with low-frequency stimulation that can induce long-term depression. Another possibility, again, it's not mutually exclusive, but another possibility is that the precise time relations between the spikes of the presynaptic and the postsynaptic cells would define the changes in the synaptic strength. This is also called as spike time dependent plasticity, or Hebbian or anti-Hebbian plasticity. And I will quickly show two classical results. So here is long-term, is high-frequency stimulation done in the rabbit. This is before the stimulation, and it is basically the amplitude of a population EPSP. It's basically the deflection of the LFP following a stimulus. And this is what happens after repetitive stimulation. So there is some potentiation of the field effect. And this is accumulating over multiple high-frequency stimulation events over hours. Another possibility is to pair presynaptic and postsynaptic activity. So here a presynaptic pulse is generated, and also a postsynaptic spike is generated. And this also potentiates the synapse. Here it's the measure is intracellular and it's a bit different, but the concept is similar. For spike timing dependent plasticity, the critical parameter is the timing relations between the pre and postsynaptic cells. So in these types of plots, spike timing means the number of milliseconds that the presynaptic cell spiked before the postsynaptic cell. So 20 milliseconds in this abscissa means that the presynaptic cell spiked, and 20 milliseconds later, the postsynaptic cell was induced to spike. This is the manipulation. And the result is that, for example, the EPSC increased by 50% relative to its spontaneous level. Now, there are multiple open questions about these types of plasticity. One of them is whether this can be observed also at the spiking level, not only at the level of EPSPs. The other question is whether they can be observed in the intact brain, so not only in cell cultures or slices. Another question is a question of specificity. So it is unclear whether Hebbian plasticity applies only to excitatory cells, or also to excitatory to inhibitory cells, or whether these types of synapses are anti-Hebbian. So this is what we set out to determine. Again, in freely moving mice using multi-site optical stimulation. This project is spearheaded by Lido, and the protocol is to record from the freely moving mouse for about half an hour, sometimes more, then to apply a manipulation for about 45 or 50 minutes, and then repeat the recording of the without any manipulation. And what we do is we compare the correlations or the precise timing between the spikes of the pre and postsynaptic cells during the post-light stimulation with the patterns before light stimulation. So this is just a schematic. We use two approaches. One is feed-forward pairing. We use this in CAM-KNAS channel rhodopsin animals in the neocortex. the idea is to pass a searchlight or a focus of light throughout the brain. So we activate different neurons at different times. And the hypothesis is that different cells that were activated earlier, presynaptic cells that were activated before the postsynaptic cells, these synapses would potentiate. So this is the way we actually do these experiments. This is 3 sites in the brain. The first light applies to these electrodes, the second light applies to these electrodes, and the third applies to these electrodes. So you can see that we're generating a sequence of, this is multineuronal spikes, spike trains throughout the brain. This is how 70 simultaneously recorded neurons look at a time scale of about 50 milliseconds. And this is an example of 2 pyramidal cells in the neocortex in which one cell, on average, spiked before. Now, how do we analyze these data? We compute the cross-correlation histograms normalized to spikes per second per trigger spike before the stimulation and after the stimulation. And we compute the gain in the synaptic activity, that is the area under the curve here, which is above the chance level line, before and after. So this is before the stimulation, and this is after the stimulation. You can see that the integral here decreased. And indeed, the stimulation was such that the presynaptic cell was activated after the postsynaptic cell. So the synapse or the timing relation between the cells were such that under Hebbian mechanism, the synapse or the connectivity would be reduced, and indeed it was reduced. When we accumulate, these are difficult and low yield experiments, so that we don't have too many data points yet. But if we look at pyramidal cell to pyramidal cell synapses and we plot the change in the spike transmission gain, that is the difference between the integrals divided by the sum, as a function of the time lag, that is the peak in the cross correlation between the PSTHs, we see that most of the pairs in which the presynaptic cell was activated before the postsynaptic cell potentiated, whereas most of the cells that the postsynaptic cell was activated before the postsynaptic cell were depotentiated. So most of the points are in the first and third quarters. The last part that I would like to share with you is what happens when we try to focus on pyramidal cell to interneuron synapses. So to do that, we use another type of animals, which is PV channelrhodopsin mice. Here we do the experiments both in the neocortex and CA1. In CA1, there are not too many pyramidal cell to pyramidal cell connections, so the first type of experiments are not too relevant. But when we use, here we use a closed loop pattern. So in real time, we detect the spike and sort the spike of a single pyramidal or a small group of pyramidal neurons. And we stimulate the PV interneurons after we detect this spike. So we can only work here with positive time lags, but we have the cellular specificity. This is how the experiment looks like. This is just 3 out of tens of channels that we use, but for demonstration, this is the time of the presynaptic spike or the real-time detected event. It's here. And then we apply light and induce spikes in the postsynaptic cell. So this is a pyramidal cell, and this is an interneuron in CA1. These are the PSTHs of many simultaneously recorded cells, and this is of one pair. And when we do this, what we see is that, again, there are not too many data points here, but the results are significant, actually, in terms of most of the points being in the first quarter. Now, they cannot be in the second or third quarter, but they can a synapse can either potentiate or depotentiate, and most of the connections potentiate. Again, this is for close by pairs. Most of them are several tens of micrometers apart, the pyramidal cells and the interneurons, and some of them are up to 400 microns apart. So to answer this question of what changes the strength of a synapse, I would say the following. If it's an excitatory connection between a pyramidal cell and another pyramidal cell, then between spikes in freely moving animals, we observe Hebbian plasticity. So it's full bidirectional Hebbian STDP with LTP and LTD. On the other hand, for the connections between pyramidal cells and interneurons, we do not have sufficient information about this part for the same cells. But what we can say is that LTP in these cells is Hebbian. So if a pyramidal cell spikes before an interneuron, the synapse between these two will also potentiate. So to summarize, and I'm a bit after time, I'm sorry, but we started a bit later, so let me just summarize. We asked two questions. What makes a neuron spike and what changes synaptic strength? We used a novel approach of manipulating individual spikes or small groups of spikes of cells in the brain of live, freely moving live animals to answer this. And what we find is that most of the indirectly activated neurons, that is the synaptically activated cells, behave as temporal coders. And with respect to the second question, we find that synaptic strength changes in a Hebbian manner, both for connections between excitatory cells and excitatory cells in the neocortex, and between excitatory cells and inhibitory cells in the hippocampus and the neocortex. Thank you very much for giving me the opportunity to give this talk. These are again the people that did the work. This is the funding, and if there are any questions, I would be happy to answer.
So to avoid the problems we had with the sound, I will ask Dr. Stark to read the question himself. So hopefully he will read the questions and answer them.
So I think what I should do is go to the ask the question and see what people ask. So one question is, I will go by votes. How do you determine which are the presynaptic cells and which are postsynaptic cells in your experimental protocol? So the answer to this is this graph. So in the pre-stimulation, that is the baseline condition, we collect data for about half an hour, sometimes up to 2 hours. So we have the pattern of connectivity or the spike timing of the two cells, or any two cells that is, in the lack of any manipulation. And we compute a cross-correlation histogram between these two. So I assume that whoever is asking the question understands how to compute the cross-correlation histogram between 2 point processes. Now, once we do that, we are looking or we are asking the statistical question of whether these two cells are connected or have a synapse between them, or a set of synapses between them. And the way we do this is we use a jittering-based predictor, which is essentially to convolve this histogram, this pink histogram, with a window. We obtain, this can be done in multiple manners by spike time jittering or interval jittering or a convolution-based method, but the essence is very similar. We obtain a predictor, which basically means what are the timing relations between the cells, given the fact that spike timing does not matter up to a window of, let's say, 5 milliseconds. Then we ask, what is the probability to obtain this count when this number is expected? And we get a p-value, which we correct statistically for multiple comparisons, etc. And if we have an extreme p-value, then we say the two units have a synaptic connection between them. And by the time lag of the peak, we can say which cell is presynaptic and which is postsynaptic. And by integrating this curve, the area above the chance level, we can also quantify the strength or the gain of the synapse. I hope this answers the question. So this also answers the second or related question of how did you determine that they are paired? A second question is how do you take into account the noise from turning on the LED and movement artifact from freely behaving mice? So this is a very important technical question, which I will be happy to explain on a one-to-one interface. This is a lot of the work in this field is indeed to get very clean recordings. The LED, I will just say in briefly, the LED or the light or the electrical artifacts from turning on the LEDs are removed by shielding and grounding. And the same approach is used to minimize movement artifacts. We use dereferencing and grounding and filtering techniques. So it's a whole issue. The third question that I can see here is, does the optical stimulation of the brain influence the behavior of the animal in any way? So yes, of course, we can talk to the animal through stimulation of the brain. We can induce different patterns of neuronal activity in the brain that will cause the animal to behave differently, to make different choices, to learn. But this is not what happens in these experiments. In these experiments, all the stimuli are very weak, they're very localized, and we do not, in these experiments, ask questions about the behavior. So the animals are not involved in some behavioral task during these experiments. They're just in the home cage. We monitor their position, their acceleration, various parameters, and we don't see a specific change in these patterns during or before compared to after the stimulation. But the answer to the question of does optical stimulation in the brain influence the behavior of the animal in any way? So the answer to that is certainly yes. I don't know if I have time for any more questions. So I will just continue answering questions until somebody tells me otherwise, or should we stop? Okay, I see that. Okay, I will just continue. How would this change in response, i.e. pyramidal to pyramidal stimulation, may be related to sparse coding? So what we see is that different pairs of pyramidal cells that are recorded simultaneously can change their synapses in different magnitudes and directions. I will rephrase. We have 4 pyramidal cells that are recorded at the same time. Each 2 have, let's divide them into 2 groups of 2, and they have synaptic connections. By making a sweep of light, a specific pattern of activation, we can generate a situation in which the connection between 2 of these cells will increase and 2 others will decrease. or the effective connectivity, or the synaptic or the spike transmission gain will increase in one pair and decrease in the other. So I think this would be a part of the definition of sparse coding. The last question that I think I did not answer is how do you determine connections between excitatory cells given the very low transmission probability? So again, I think I answered this in the statistical part. So I think I'm out of time. The clock is 45 minutes, and I don't want to take any time from anybody else. So thank you for listening and asking.